Happy after lunch, everyone. We're about ready to get started. We're at the 1:31 mark. We're already behind time. I'm Ed Prysdale. I'm a scientist for Canadian Blood Services. I have a clinical pathology professorship at the University of British Columbia, and I am the Associate Director of the Center for Blood Research. And I'm thrilled to be introducing speakers this afternoon. One little line of business is that I also have the dubious distinction of timekeeper, so speakers, keep it going, and this is what happens when you're really in trouble. High tech, but highly effective, Cedric. I am thrilled. I'm getting the finger shake from the directors. It's an okay finger shake. So I'll just ham it up a little bit longer. Yeah. Cedric's giving me the finger too, I see. One more minute. So what do we want to talk about here? We're having some photo opportunities. Maybe we should get Jean in here. Where is she? It's wonderful to have so many people going on. Let's start. I don't care what he says anymore. One of the most inspiring conversations I've ever had, believe it or not, revolved around a kid's book at one of the UBC bookstore. And the book was about Darwin's expedition on the HMS Beagle. And it was one of these pop-up books. So the person I was having this discussion with and I were sitting on this chair, and there it was. The discussion started evolving. I'm going to go slower, Conway. Wait a second.
Cover your eyes.
Cover your eyes, cover your eyes. There we go. So the conversation eventually evolved around unicorns and whether they actually existed. And I'm thrilled to be introducing the speaker today, the person that I had this discussion with, and that's Professor Edmund Fischer, and who in 1992 shared the Nobel Prize for Physiology and Medicine for the seminal discovery of protein phosphorylation as a molecular switch in cell physiology. Eddie was born in Shanghai, and when he was 7, his family moved and settled in Switzerland, which is where he eventually obtained his PhD in chemistry. As a side branch, Eddie performed as a concert pianist, very left and right focused. His first and only tenured academic appointment was at the University of Washington in Seattle, where his phosphorylation studies were conducted. And, well, without further ado, I am thrilled to introduce Eddie to give a talk.
Well, it's really a very great pleasure for me to have been invited to talk to you today, all the more that I know Earl for more than 60 years, and we have been all over the world together. And here's a picture of Earl taken in France at a meeting in Annecy, and then we've been many times in China. This is a Zhangjiajie. We're together there, and also here, and finally at a ceremony in Changsha, where we were given an honorary professorship at the Hunan University. Okay, and thank you for the invitation, by the way, to be here. What I will do today is to give you a very, very rapid overview of the work we carried out with my friend Ed Krebs on the enzyme glycogen phosphorylase, work that led to the discovery of reversible protein phosphorylation. As you will see, it is an embarrassingly simple reaction, and nobody would have paid any attention to it if it was not for the fact that it turned out to be absolutely crucial for the regulation of cellular processes. On the other hand, it's fair to say that when we came up with it, it came up as a very big surprise, because 65 years ago, nobody knew anything about cellular regulation or enzyme regulation. The prevailing idea was that enzymes were regulated by the rate at which they were synthesized and then degraded. And that stood until the mid-40s, early 50s, when it was realized that this could not be the case, that this wouldn't work, that cells had to have the possibility of manipulating the activity of the enzymes once they had been produced inside the cell. They had to have the capability of adapting to the environment, of satisfying their physiological needs almost instantly in response to whatever demand was placed upon them. And that was really one of the major problems that faced the biologists in those days, and that's precisely the problem that we decided to investigate with Ed Krebs. We wanted to know how the activity of an important enzyme, glycogen phosphorylase, was regulated. In fact, very important, because phosphorylase was known to catalyze the first step, the rate-limiting step of carbohydrate metabolism, to give blood glucose in the liver. By the way, that is the most important function of the liver, or ATP everywhere else, in every cells. Phosphorylase also was known to be under complex hormonal regulation, being activated by adrenaline and inhibited by insulin. When it was discovered in the early 30s by Parnas in Poland and Carl and Gertie Corey in the United States, the enzyme was known to exist in a totally inactive form in the muscle, and to have an absolute requirement of AMP for activity. And automatically the Coreys assumed that AMP would serve as a coenzyme for phosphorylase. And that stood until the very early 40s, when a gal in Corey's lab, Arda Green, succeeded in purifying the active form, but she obtained it in crystallizing the enzyme, but she obtained it in a new form, in that it was totally active in the absence of AMP. So they called that new form phosphorylase A, and logically assume that it had to contain AMP bound covalently to the protein as a prosthetic group. And they further assumed that that had to be the native form of the enzyme in muscle. Because if they let muscle extracts standing for a few, not even an hour, it was rapidly degraded, they thought, to the old form that they now called phosphorylase B, the form that required AMP for activity. So an absolutely logical hypothesis that was accepted by everybody, except that they could find no AMP produced in that reaction. And utilizing the most sensitive analytical procedures available, they could find no AMP in the native enzyme. So there the Coreys were stuck. They knew that the enzyme existed in two forms, but they did not know in which way those two forms might differ. And strange as it would seem today, they actually dropped the problem. They were working on phosphorylase for 10 years. They had a big group. They dropped that problem. And that is precisely the problem that we decided to investigate with Ed. Here's a picture of Carl and Gertie Corey when they got the Nobel Prize in 1947 for the discovery of phosphorylase and their work on carbohydrate metabolism. Okay, so there we were, and the first thing we had to do, of course, is to isolate the native enzyme phosphorylase A in great amount. So we cleared the bench in the lab and started working, just the two of us, side by side, utilizing the classical procedure of Corey's to prepare native phosphorylase A. Now, Corey's preparation consisted in taking rabbit muscle, grinding it in a meat grinder, extracting it, the mess in the hamburger, in water for about an hour, throwing the gunk in a big cheesecloth, squeezing the cheesecloth like crazy to get very turbid first extract, and then clarifying this first extract by passing it through a battery of large filter papers. Of course, the filter paper would get clogged up right away, so they took a big glass rod, poked a hole in it, and went from one filter paper to the other. And it wasn't unusual for them to utilize, 15, 20 filter papers in one preparation. As you can see, the method was absolutely archaic. And in fact, here's a picture of our lab with Ed. Okay? Ed is standing there under the top hat. Anyway, so let me go back. So we decided with Ed, enough with that jazz. Let's replace the paper filtration by a centrifugation. In Corey's defense, it should be said that in 40s there was no large capacity centrifuge, but as fast as we could work, as cleanly as we could work, when we centrifuged our preparation, we could never obtain active phosphorylase A, only the so-called degraded B form. Until finally, in desperation, we said, this is idiotic. The system works for the Coreys, it doesn't work for us. What the hell is going on? We have to go back to Corey's procedure, filter paper filtration and all, measuring every step to see what the hell is going on. And to our immense surprise, this is what we found, that the very first extract from the muscle did not contain active phosphorylase, but the so-called degraded B form. And yet, when that solution, the suspension, was passed through filter paper, it became activated to phosphorylase A. Now, speaking of a disappointment would not be the right word. Two guys dreaming to find a very sophisticated mechanism of activation, a very great chemical reaction, a cofactor, a new hormone, paper filtration, to activate the enzyme was embarrassing. It was really below the worst. It was the pits. But when we investigated what was happening, we found out that it was not the filter paper that carried out the conversion, but the fact that all filter papers, probably still today, were contaminated by absolutely trace amounts of calcium ions. But enough were picked up during those 15, 20 filtrations to do the trick. And then rapidly we found out that calcium did not act alone, that ATP always present in fresh crude muscle extract was also required, and that immediately suggested we must be dealing with a phosphorylation reaction. Now, in those days, you had no ATP32. You had to synthesize the damn thing yourself. But we knew that Art Kornberg, who was in St. Louis, had prepared some ATP32 that he needed for his beautiful work on the biosynthesis of DNA. So we called Art, and he immediately sent us a sample of gamma label ATP32, with which we could show that the radioactivity was introduced in a protein fraction that we could isolate and which turned out to be phosphorylase. So for the first time, we could propose that the regulation of phosphorylase occurred like so, that inactive phosphorylase B was phosphorylated and activated in a reaction that required calcium, magnesium ATP, and an enzyme, of course, which we call phosphorylase kinase, and then the reverse reaction had to be catalyzed by a phosphorylase phosphatase. So a reversible phosphorylation reaction. And we shipped immediately the manuscript to the Journal of Biological Chemistry, to the JBC. Now, in those days, the JBC had a very small editorial board. I think there were no more than 5 or 6 of the top biochemists in the country that served on the editorial board. And we knew that Carl Corey was one of the editors of JBC. So, no, we had faith that he would not take advantage of the situation, and he absolutely did not. In fact, Carl was very excited by our data, and he never touched that problem. Here's a picture of him, Carl Corey, that I took in 1965 when he was visiting, and I took him for a walk at the foot of Mount Rainier next to Seattle. Well, the reaction we found by today's standard is really enormously simple. But when we began to purify the elements of the system, we had succeeded crystallizing phosphorylase B, and we had partially purified phosphorylase kinase. It was clear that calcium did not act in that reaction. And yet, as I just told you, it was absolutely required when crude muscle extracts were used. In crude muscle extracts where you have a lot of phosphorylase B, a lot of phosphorylase kinase, a lot of magnesium ATP, no conversion takes place until you add calcium. So the only possibility was that calcium had to act at an earlier step, and the possibility was that phosphorylase kinase, just like phosphorylase, also existed in an inactive and active form, and perhaps calcium was required for that reaction, and that turned out to be correct. In the presence of calcium ions, phos kinase is rapidly phosphorylated, and then it could activate phosphorylase. So an enzyme that can participate in the activation of another enzyme. Now, in those days, one knew very little about the mode of action of hormones. One knew that hormones would act in the cell membrane, but what happened next was totally unknown. It was unbelievable that a hormone could act on a cell-free system until Earl Sutherland and his student Ted Roll discovered cyclic AMP that served as a second messenger for the activity of adrenaline. So Earl sent us immediately a little bit of his cyclic AMP, and we could find that it accelerated greatly that reaction, either by increasing the rate of phosphorylase kinase, autophosphorylation, or perhaps by acting on yet another enzyme, for which, as a better term, we call a kinase. Now, when we sent the manuscript to the JBC, the reviewer who examined that didn't like that at all. First of all, he didn't like the idea that an enzyme would act on an enzyme. And to suggest that a third enzyme might be involved, he wrote, "When will it end?" Furthermore, he said, if indeed a third enzyme is involved, I wish that the authors could think of a better term than a kinase. Little did he would have known that, a few years later, the MAP kinase pathway was discovered, where you have MAP K, MAP KK, MAP KKK acting successively on one another. Anyway, that hypothesis was confirmed 4 years later by the isolation of the cyclic AMP-dependent protein kinase, and since Earl and his group had clarified the production of cyclic AMP at the membrane, the whole pathway for the degradation of glycogen was resolved. We did not know in those days if that phosphorylation reaction was restricted to these three enzymes or if it was a more general reaction. As luck would have it, it turned out that as you know, that protein phosphorylation is one of the major ways by which metabolic reactions are being involved in the control of metabolism, transcription, translation, the immune response, cell differentiation, transformation, secretion, etc., cell death. It's involved in many hereditary diseases or pathologies, such as, of course, diabetes, but also directly involved in Alzheimer, in Parkinson's disease, in chronic myelogenous leukemia. It's involved in bacterial diseases such as cholera and the plague. By the way, the virulence factor of Yersinia pestis, the agent of bubonic plague that wiped out practically whole populations is a tyrosine phosphatase. The bug injects that tyrosine phosphatase in the host cell, where it brings about a catastrophic set of tyrosine dephosphorylation, which wipes out the immune system of the cell. And viral diseases such as smallpox, and very prominently, of course, in cancer. We were often asked with Ed if we realized at the beginning that we were dealing with a very ubiquitous and therefore very fundamental reaction. Absolutely not. We thought it was a nice reaction. We knew it was an important reaction, but we absolutely never could have guessed all the developments that follow. I have been often asked, how was it to work with Ed for 45 years? Well, I can tell you it was very easy. It was very, very enjoyable. At first, Ed was always make fun at the way I spoke English, because every morning I said, let's go take a coffee, because in French you say prendre un café. So he was making fun of that. What Ed didn't realize is that in the following months, while my English didn't improve very much, his deteriorated completely. Anyway, those were really marvelous, marvelous years. What we did was never work for us. We never considered what we did as work. It was really fun. Thank you for listening. Sorry? Yeah, sure. But you have to be next to me because my ears are lousy.
Are there any questions? Thanks very much. Eddie, sir, you are a hard act to follow. But that's exactly what our next speaker's got to do. But that's okay. I'm really excited to invite the second recipient of the 2017 Earl Davie Symposium Award, Professor James Morrissey. He's really an all-around nice guy, no question about it. Over the past couple of months, Jim has moved his lab from the University of Illinois in Urbana to the University of Michigan in Ann Arbor. No small feat, as some of you may know. He did his PhD at the University of California, San Diego, and later postdoced at Oxford and at Scripps Clinic. He has made so many contributions to understanding the initiation and regulation of blood coagulation. I've seen an abridged version of his CV, and he has a list of accolades. Well, it's kind of like reading War and Peace. It's huge. He is certainly deserving of Earl's Award, and he will be telling us a little bit about some of the contributions he's made toward regulation of blood coagulation.
Well, thanks, Ed, for that incredibly kind introduction, and I'm really deeply honored to be here to celebrate my colleague's wonderful career. And I've spent the last 30 years of my career focusing on the biochemistry of the blood clotting system, and it was definitely standing on the shoulders of giants like Earl Davie. So delighted to be here. So as the title suggests, I'm going to talk today about some of the research we've been doing on polyphosphate and its role in hemostasis and thrombosis and beyond. But I thought I'll start actually by attempting to thank the people. This slide was made in a PC and now it's being showed on a Mac, so with obvious connections there. But these are the folks who really I need to credit for the research that I'm going to talk about. And I'd really like to point out in particular Stephanie Smith in my group, who was involved in the polyphosphate story right from the beginning, and also Roberta DeCampo at the University of Georgia, who introduced us to polyphosphate. And before I talk about the polyphosphate studies, I thought I would just maybe step back and talk a bit about the coagulation cascade. It's, I think, the 53rd anniversary of the waterfall hypothesis that Earl and colleagues presented back in 1964. And it still looks much the same, although if you saw that waterfall hypothesis, tissue factor was nowhere to be seen on that. And we now know that it plays a really important role in the clotting system. So I thought about what do we know about the clotting cascade in both hemostasis and thrombosis, and what remains to be understood. And in particular, is it always the same? Is it always triggered in the same way in normal hemostasis versus thrombotic disease? It's certainly been a holy grail of our field to identify drugs that would be antithrombotic without bleeding side effects. And there's some glimmer of hope now that that might be possible, although almost all the existing drugs, in fact all the existing drugs on the market, target this final common pathway that you see here with really the potential for devastating bleeding side effects. Okay. So how is clotting initiated in normal hemostasis? Just sort of in broad terms, we now know that it's triggered by tissue factor. If you cut yourself, what's going to stop you from bleeding is tissue factor being exposed to blood. This is the physiologic aspect of the clotting cascade. And an important concept to my mind is that it relies on pre-existing triggering molecules that are positioned all around the vasculature. The reason is that it has to happen on very rapid time course. there's no time left when you cut yourself to synthesize any new molecules. You have to trigger clotting on a time scale of seconds. And so those triggers have to be pre-existing and positioned everywhere in your body so that that cascade can be triggered. And once we cloned tissue factor back in the mid-1980s and isolated monoclonal antibodies, we determined the anatomic location of tissue factor, and one of the most striking things was that it's really abundant in the adventitial cells that surround all blood vessels larger than capillaries. And of course, in the subendothelial space, there's abundant collagen, which is an excellent trigger of platelet function. So if you break a blood vessel and blood attempts to leak out of the vessel, it's going to come into contact with tissue factor and things that activate platelets like collagen. Those are pre-existing molecules that have been sequestered from the flowing blood until you have vascular damage, and you're going to trigger a clot. Okay, so that seems very clear-cut. That's how tissue factor functions. And this is just an example of the distribution. So in red is tissue factor, showing what Tom Edgington, I think, quite nicely called the hemostatic envelope around the vasculature, and really there's abundant tissue factor in many epithelial cell layers around in the body, and it's especially abundant in places that would be devastating to bleed. Okay, that makes sense. How is clotting triggered in thrombosis and also in coagulopathies like sepsis that we've heard about before, earlier today, but also other life-threatening coagulopathies like trauma-induced coagulopathy. So simple crush injury in the absence of any infection can lead to a life-threatening coagulopathy. What's triggering the clotting cascade in those cases? And what do we know about that? Well, an important concept that's been put forward recently by folks like Engelmann and Mossberg and other colleagues, is the notion of immunothrombosis. So it's been known for many years that fibrin deposition is one of the hallmarks of inflammation. The induration that you see in say, skin inflammation is due entirely to extravascular deposition of fibrin. And there are a whole series of studies that have shown, I think quite elegantly, that infection by organisms such as Yersinia pestis, plague organism, rely on evading this host response of walling off infections with fibrin. So the clotting cascade, in addition to its role in hemostasis, clearly is an effector arm of the innate immune system. So we can think of the activation of the clotting in these kind of situation as a pathologic aspect of the clotting cascade, part of the host response to pathogens, an effector arm of the innate immune system, and those triggers are evolved on a much slower timescale. So the fastest you can get a monocyte to synthesize tissue factor is something like 3 or 4 hours. And probably in some cases you're elaborating these activators of the clotting cascade in the context of inflammation on a time scale of hours to days. So the time scale certainly is very different, or can be very different, in thrombotic diseases compared to hemostasis. And it's possible that the triggers are different. Now, in this interesting paper in Nature Reviews Immunology, Engelmann and Mossberg point out that this immunothrombosis is normally protective. We have it as a part of the inflammation that's needed to fight infection. But it can also go awry, and particularly if it's activated systemically. It's extremely bad for us. Now, what are the triggers of the clotting cascade in immunothrombosis? Tissue factor possibly in many cases, but are there other triggers? And that's a subject of really intense investigation right now, is what are those other triggers? People are focusing on PAMPs and DAMPs, so pathogen-associated molecular patterns or damage-associated molecular patterns, various molecules that are being released from infected cells or microorganisms that can do things to the innate immune system, including activating the clotting cascade. Now, we know a lot about how the clotting cascade is triggered in myocardial infarction, probably the best studied and best understood type of thrombotic disease. And that's thought now to be largely driven by tissue factor. How does that happen? Well, atherosclerotic plaques are infiltrated by not only monocytes, which differentiate into macrophages/foam cells, but also vascular smooth muscle cells infiltrate into this area. And both of those cell types normally express very little tissue factor, essentially undetectable amounts. But when you activate them and they get activated in a plaque, they express a lot of tissue factor. And you can see that if you section an atheroma and stain it for tissue factor. The necrotic core has a lot of active tissue factor, but also the cellular margins have a lot of tissue factor in them too. And we now know that plaque rupture or plaque fissure exposes basically these tissue factor containing cells directly to the flowing blood, and you can get a blood clot right away. And so there's two aspects of the timing here. It took you probably a very long time to generate the tissue factor in these plaques. But the reason that it has a very fast time course once you break a plaque open is that then you get this prompt activation of the clotting cascade. Okay. But it's tissue factor that trigger in all types of thrombosis. And the answer there is less clear, I think. But there's more and more evidence suggesting that there are interesting other molecules that are participating in thrombosis in addition to tissue factor that can trigger the clotting cascade. And in particular, there's been a resurgence of interest in the contact pathway, the pathway that we saw in that waterfall hypothesis as the one that's driving the clotting cascade. So what are the known activators of the contact pathway? Well, for many years, they were anionic surfaces, things like glass, clay, diatomaceous earth, and this peculiar chemical, ellagic acid. And in fact, if you perform an APTT assay, clotting assay, you're going to basically add a ground-up version of one of these things, like silica particles or clay or diatomaceous earth or ellagic acid as a trigger of the clotting cascade. Now, a lot of people have wondered what are the real pathophysiologic activators. It's certainly likely that we evolved this pathway to interact with other molecules other than things like dirt and glass. But what are those real pathophysiologic activators? There are several interesting candidates that have been put forward recently. These include extracellular RNA, as Patricia pointed out, extracellular DNA, either through necrotic cells releasing DNA or nets elaborating DNA. And more recently, we've shown that inorganic polyphosphate, especially long-chain inorganic polyphosphate, is a really potent activator of the contact pathway. And other folks have recently shown that certain unfolded proteins can also activate the contact pathway. So I'll focus my attention for the next few minutes on polyphosphate, the molecule that we've been studying really for the last 11 years as a activator of the contact pathway and a modulator of downstream clotting reactions. So what is polyphosphate? Well, it has a very simple structure. They're simply linear polymers of inorganic phosphate, and they're connected by high-energy phosphoanhydride bonds, the same bonds you'd see in ATP. So if you imagine the three phosphates in ATP, if you were to extend that chain until it was hundreds or even thousands of phosphates long, you would have polyphosphate. Polyphosphate is ubiquitous throughout life. Probably every organism on the planet makes it. It's been studied most extensively in microorganisms, so we understand much more about its metabolism and roles in microorganisms than we do in higher organisms. Polyphosphate tends to be stored in granules, and this is true even in bacteria. In higher organisms, eukaryotes, those granules that are generally called acetylcalcisomes. They're called that because they're a membrane-bound organelle that contains a lot of metal ions. They're always acidic, and the major counterion to those metals is polyphosphate. And it makes sense if you think about it. If you need to pack a lot of metals in a very small space, having a polymerized counterion makes sense because the osmotic effect of a polymer is far less than the osmotic effect of the same concentration of phosphate monomers. In addition, cells really need to control the concentration of phosphate monomers, so that's not an option for them either. If you do electron micrographs of sections of these cells, the acetylcalcisomes stain black, right, because they bind heavy metals very easily. Now, just by chance one day, Roberto de Campo, who used to be at the University of Illinois when I was there, looked at an old electron micrograph of a human platelet, and he was struck by the fact that they contain these electron-dense granules that looked a lot like the electron-dense granules in trypanosomes that he was studying. He mostly focuses on this particular trypanosome that causes Chagas disease. He did some reading about platelet-dense granules and found that they were filled with these same kinds of metals. They're also acidic, membrane-bound compartments, but no one had ever looked to see if they had polyphosphate as the counterion in them. So he got a unit of expired platelets from the blood bank. He isolated the dense granules and extracted them, and what he discovered was that they were full of polyphosphate. And furthermore, if you stimulate platelets, they secrete all their granule contents. They secrete the dense granules, they secrete the alpha granules, they even secrete their lysosome contents. And this included the polyphosphate. Interestingly, if you look at polyphosphate in almost any microorganism, it's extremely heterogeneous in size. So the polymer lengths range from anything from maybe about 10 phosphates long all the way to over 1,000 phosphates and everything in between. But remarkably, the polyphosphate that was packed into dense granules had a very narrow size distribution of about 60 to 100 phosphates long. Now, in contrast to microorganisms, no one has discovered the enzymes that are responsible for synthesizing polyphosphate in mammals. So we do not understand how polyphosphate is made and stored in these granules, and we certainly don't understand what's regulating that size length. But as I'll come back to a little later, we think that the platelets are regulating, or the megakaryocytes are regulating these polymer lengths on purpose, okay, because it controls what that polyphosphate does to the blood clotting system. Polyphosphate is relatively abundant. If you activate all the platelets in blood, you could end up with something like 1 to 3 micromolar polyphosphate. And that's always, I should point out, when we talk about concentrations of polyphosphate, those are in the field, those are almost always reported as the concentration of phosphate monomer. And the reason for that is we don't always know the polymer length of polyphosphate, but we can always hydrolyze it down to monomer and measure that. So if you wanted to get the polymer concentration, in this case you would divide by about 70. Okay, so when Roberto discovered that platelets were secreting polyphosphate, he didn't know anything about blood clotting, he didn't work on that. But he figured, well, platelets do something in clotting. They probably, if they're secreting polyphosphate, it probably modulates clotting. So he just knocked on my door one day and said, you know, I've discovered this. You guys should look at polyphosphate. And he was thinking that polyphosphate would be an anticoagulant like heparin. So heparin's another linear molecule, extremely negatively charged. It's a really potent anticoagulant. So he gave us some polyphosphate, and we did some screening assays, and this is one of the very first ones that we did. So this is just a 96-well plate-based clotting assay in which we took basically citrated plasma, put it in a 96-well plate, recalcified it. We added a little UPA to activate the fibrinolytic system. And in plastics, a very poor activator of the contact pathway, so it took 25 minutes for it to clot. We can see light scattering due to fibrin deposition, fibrin polymerization, excuse me. And then the plasmin that was generated digested the clot away. If we add polyphosphate of roughly the size that platelets secrete and low micromolar concentrations, what we found was that the plasma had clotted completely by the first time point. And furthermore, it became much harder for the fibrinolytic system to lyse that clot. So it has really the opposite effect of heparin. It's procoagulant and antifibrinolytic. And this really got our attention. So I'll summarize almost a decade's worth of work in this one slide. So polyphosphate does multiple things to clotting, but it doesn't do any, it doesn't affect all the reactions. It's specific. If you have really long-chain polyphosphate, it triggers the contact pathway. And in fact, on a weight basis, it's by far the most potent known triggering agent of the contact pathway. The second thing we discovered is that it greatly accelerates the rate at which factor V is converted to 5A. And factor 5A sits at a really important sort of junction point of all of the clotting cascades. And something that I really hadn't anticipated before doing these experiments, but if you change the kinetics at which factor V gets converted to 5A, you really have some very interesting effects on the clotting system. And in fact, you render clotting largely insensitive to protease inhibitors of various kinds. I'll come back to that very briefly later. The third thing we discovered is that polyphosphate appears to become physically incorporated into the fibrin clot and changes the structure of the clot, makes it stronger and harder for the fibrinolytic system to lyse. And then most recently, we've discovered that polyphosphate appears to be the missing cofactor that can explain the activation of factor XI by thrombin, and I'll talk about that in a little more detail in just a second. Now, as I mentioned, if you change the kinetics by which factor V gets converted to 5A, you render clotting reactions insensitive to a whole range of anticoagulants. And this includes anticoagulant drugs like just heparin, low molecular weight heparin, even direct 10A and thrombin inhibitors are largely ineffective in the face of adding polyphosphate or simply spiking 5A into plasma. It's really quite remarkable. Now, as I mentioned, we discovered that polyphosphate changes fibrin clot structure. The turbidity of the clots is quite different in the presence of polyphosphate. This is an experiment in which we took purified fibrinogen and we clotted it with purified thrombin. And in the absence of polyphosphate, you get the typical sort of fibrin fibrils. These are these little tiny fibers that make bigger fibers and generate the three-dimensional clot. As we put in more and more polyphosphate, however, what we found was that those fibers become much thicker. They're also mechanically stronger. Now, one way that you can manipulate the fibril diameter is by changing the ratio of thrombin to fibrinogen when you make the clot. And so you can actually recapitulate some of these differences with some of these findings here, but with one huge difference. Almost everything you do to make a fibrin fibril thicker renders it actually more sensitive to fibrinolysis, not less sensitive. So polyphosphate unusually makes the fibrin fibers thicker, but harder for the fibrinolytic system to act on. And Nikki Much, who was a postdoc in my lab, who's gone on to do her own independent studies at Aberdeen, recently showed that polyphosphate appears to be interacting with fibrin in such a way that it masks the C-terminal lysine residues that the fibrinolytic proteins need to gain a toehold onto the fibrin. Now, we collaborated with Thomas Rene at the Karolinska Institute and published a paper in 2009 in which we looked at in vivo effects of polyphosphate. And if you inject long-chain polyphosphate, especially subcutaneously, it causes capillary leak. This is because it activates the contact pathway, which generates bradykinin. And he was able to show that 12 deficient animals don't have this. Neither do animals in which you knock out the bradykinin 2 receptor. And furthermore, you can actually kill animals by injecting polyphosphate intraperitoneally, and the 12 deficient and bradykinin receptor animals are protected. So polyphosphate can be highly pro-inflammatory in addition to being procoagulant. Now, almost all of the experiments I've shown you up till now were done with relatively heterogeneous preparations of polyphosphate. We initially were using polyphosphate we got from Sigma, and this is run on just a polyacrylamide gel, and you can see that even the various different sizes are relatively heterogeneous. On the left here is some polyphosphate isolated from salmonella, and you can see that it's very heterogeneous indeed. In the middle here, this is polyphosphate isolated from platelets, so a very narrow size distribution. And we were wondering what was the polymer length dependence of these various reactions in the clotting system? Could that explain some of the biology of polyphosphate? And the answer turned out to be true. And so this is a summary of a series of experiments in which we looked at those various reactions that I showed you on that schematic as a function of polyphosphate polymer length using very narrowly sized fractionated polyphosphate. On the x-axis is the polyphosphate polymer length in phosphate units. I've colored in pink the range of polymer sizes that you'd see secreted by platelets, and then in yellow would be the range you might see in microbes. And the y-axis is simply the specific activity of whatever that reaction is. And we can see that activating the contact pathway, that specific activity goes up exponentially with polymer length. And so what that means really is that sort of microbial length polyphosphate is really good at triggering the contact pathway, but platelet polyphosphate is really very poor at doing that reaction. However, the downstream reactions such as fibrin clot turbidity, factor V activation, abrogating TFPI anticoagulant activity, and this back activation of 11 by thrombin are all well supported by platelet-sized polyphosphate. So if we put all that together, what it says is that microbial polyphosphate can do all of these things, but platelet polyphosphate really is good at the downstream reaction, changing the kinetics of 5 activation, supporting 11 activation, to some extent enhancing fibrin clot structure, but not really activating the contact pathway. Now let's turn our attention just for a moment to the question of factor 11. In the classic waterfall scheme, factor 11 was activated by factor 12A. The conundrum here is that factor 11 deficiency, severe 11 deficiency, is associated with a bleeding tendency. In fact, it's sometimes called hemophilia C. But factor 12 deficiency is not. So humans or animals who completely lack factor 12 have no bleeding problem whatsoever. So this said that in normal hemostasis, factor 11 has to be being activated by a protease other than 12A. Just has to be. But what is the protease? Is it something outside the clotting cascade or what? Well, in 1991, there were a couple of papers published independently by Galani Ambrose and Naito and Fujikawa, who noticed that that thrombin, the last protease in the clotting cascade, could feed all the way back and activate factor 11 to 11A. 11A then activates 9 and you propagate the clotting cascade. And both sets of authors proposed that this could create an extended feedback loop that might generate thrombin in special places that you really need it. And factor 11 deficient patients bleed under only certain circumstances, like after injury or surgery, especially in tissues with a high fibrinolytic potential. Now, almost immediately these papers were criticized on the basis that the kinetics of this reaction are extremely poor and it takes unrealistically high amounts of thrombin to make this reaction happen. And people pointed out that thrombin has lots of much better substrates to distract its attention in plasma, and that the rate of 11 activation is going to be trivial under the kind of conditions that you can imagine. Well, we wondered if polyphosphate might be the missing cofactor, especially after we discovered that polyphosphate bound with nanomolar affinity to both factor 11 and thrombin. So we did this experiment. We took the plasma concentration of factor 11, which is 30 nanomolar, and really quite a low concentration of thrombin, 5 nanomolar. And then we looked at the rate of factor 11 activation to 11A. And in the absence of any kind of cofactor, this rate it's not actually zero, but it's really slow. If, however, we add polyphosphate to this reaction, what we find is that there's a tremendous acceleration of the reaction. In fact, it enhances the second-order rate constant by about 3,000-fold. Furthermore, we typically get this bell-shaped concentration dependence, which is very typical of a template mechanism. And so what we think is happening is that polyphosphate recruits both 11 and thrombin to into the local vicinity where they can interact with each other. And furthermore, this reaction is supported really well with platelet-sized polyphosphate. So we did an additional experiment in which we took platelet releasate. So this is what you get when you stimulate platelets to secrete all their contents. We did this with TRAP, which binds to the thrombin receptor. And then we centrifuge out the platelets and just take the supernatant. So that's the releasate, everything that came out of the platelets. And we did the same reaction. And what we found was that the releasates could also greatly accelerate the rate of 11 activation by thrombin, tremendously so. If we boiled the releasate and then cooled it to room temperature, it worked great. In fact, it worked a little better after we'd boiled it, probably because we denatured all the competing polyphosphate binding proteins. And if we inactivate the polyphosphate in the releasate, either by a polyphosphate binding protein, that's PPXBD, or recombinant yeast exopolyphosphatase, which digests it down to monophosphate, that destroyed the ability of the releasate to support this reaction. So we think that actually one of the most interesting and potentially physiologically significant thing that platelet polyphosphate does is provide the missing cofactor for 11 activation, which can maybe explain the otherwise very puzzling contributions of factor 11 to normal hemostasis. So we could revise the clotting cascade to capture all the hemostatically relevant clotting factors by drawing it like this. Okay? We'll leave out most of the contact pathway because we know that that's not required for hemostasis. 11 is. Polyphosphate may be the missing cofactor that ties that all together. So hemostasis likely looks like that. Now, factor 11 is a really intriguing molecule. As I said, factor 11 deficiency has a mild bleeding diathesis, but studies done by Uri Seligson and others have shown that severe hereditary factor 11 deficiency protects significantly against venous thromboembolism and ischemic stroke. Interestingly, not myocardial infarction. And it suggests that factor 11 is participating in these thrombotic mechanisms in some way, in particularly venous thromboembolism and ischemic stroke. Probably it's not needed for MI or doesn't participate significantly for MI because it's largely tissue factor driven and very promptly tissue factor driven. That's speculation. On the flip side, if you look at general population studies, elevated factor 11 is a risk factor for venous thromboembolism and ischemic stroke. Again, not much relation to MI. Because of that, factor 11 has become a tempting target for novel antithrombotics with reduced bleeding side effects. And in fact, there was a recent, very successful initial clinical trial using antisense oligonucleotides to target factor 11 in postoperative thrombosis. So if we draw the clotting cascade to include the contact pathway and look at, on the left, the known contributions to hemostasis, and on the right, what we know about the contributions to thrombosis, you can completely eliminate prekallikrein or factor 12 and not have a bleeding problem. This is true of humans that lack these proteins, and it's also true of mice in which these proteins have been knocked out. Factor 11 deficiency gives you a mild bleeding diathesis. If you touch anything down here, you run the risk, if you overdo the inhibition, of severe bleeding. On the other hand, it's clear from human and mouse studies that you can protect against thrombosis by going after anything in the final common pathway. You can protect against at least some kinds of thrombosis by going after factor 11, not MI, but other types. And there's intriguing animal data suggesting that factor 12 can play a role in thrombosis, most particularly in the presence of artificial surfaces, but even other types of thrombosis models in mice, the mice are protected if they're factor 12 deficient. Now the case in the humans is far less clear, and I've looked over the clinical studies as of other people, and really it's a mixed bag. So I think the jury is very much out as to whether factor 12 is really important in thrombotic disease in man, and whether it would be a good target except in the case of artificial surfaces. I think that's very clear. So going after factor 12 could be very helpful in extracorporeal circulation. But whether it plays a really important role in in sort of spontaneous thrombosis remains to be seen. Now, when we saw that especially long-chain polyphosphate could trigger the contact pathway and that polyphosphate was an accelerant for some of the downstream reactions, but didn't seem to be absolutely required for those reactions, we asked the question of whether polyphosphate could be a drug target with maybe a wider therapeutic index than others. So we published this paper in 2012 in which we had done a screen and identified a number of polyphosphate inhibitors and used mouse models of thrombosis. In this particular one, it's a ferric chloride-induced carotid artery injury model. And what we found was that some positively charged molecules like some cationic PAMAM dendrimers, polyethylenamine, and the antibiotic polymyxin would neutralize the clotting activity of polyphosphate and did protect to some extent against thrombosis. The problem was all of these positively charged compounds, if you inject them into mice, they have toxicity issues, and that limited the dose that we could use and therefore their efficacy. About this time, I got to know Jay Kazakadathu here at the UBC, who was designing dendrimer-like compounds as heparin reversal agents. And these, unlike the typical cationic PAMAM dendrimers, have very low toxicity. You can inject huge quantities into mice without killing them or making them ill. He's done a couple of really clever things. The heparin binding groups are tertiary amines instead of primary amines, noticing that tertiary amines are much less toxic than primary amines. And he shielded those positive charges with PEG to reduce the affinity of the dendrimers for cell surfaces. And published this interesting paper that we helped contribute to in a somewhat small way of heparin reversal agents based on these dendrimer scaffolds as a potential basically replacement for protamine sulfate. And one of the cool things about these UHRAs is they also reverse the action of low molecular weight heparins. Given their mode of action, it seemed reasonable to us that at least a subset of these UHRAs, and I should stress that this is a large family of these dendrimer-like compounds that he's developed, a subset of them probably would be able to interact with polyphosphate and neutralize their charge and therefore their activity. So we did a screen. In this particular one, this is, I think, thrombin binding to immobilized polyphosphate, and picked some for further testing. And what we found was that these UHRAs, again in this carotid artery injury model, could be extremely antithrombotic, and with a bleeding propensity that was far less than a therapeutically equivalent dose of heparin. And Jay's lab is working on making newer generation UHRA compounds that are even more specific for polyphosphate and less likely to have general effects on the clotting cascade. So we're excited about pushing those forward. So the revised clotting cascade for thrombosis, we know that tissue factor can be the trigger in at least some kinds of thrombotic diseases. And there's interest to look at other molecules such as polyphosphate and extracellular nucleic acids as participating in thrombosis, and I'm not really going to talk about too much today, but things like trauma-induced coagulopathy. Now, interest in extracellular RNA and DNA were piqued by this interesting PNAS paper that came out in 2007 from Klaus Preissner and colleagues, in which they showed that both DNA and RNA were procoagulant, and they focused their attention on RNA because injecting ribonuclease into the animals protected them against thrombosis. So we were interested in doing a head-to-head comparison of nucleic acids and polyphosphate in terms of their ability to trigger the contact pathway. No one had really published that, and just sort of generally we got the idea that polyphosphate was a lot more active at triggering the contact pathway that nucleic acids were. On the other hand, when you activate a lot of neutrophils and elaborate a lot of nets, you're likely to elaborate orders of magnitude more nucleic acids than polyphosphate, so maybe their lower specific activity would simply be made up for by releasing lots more nucleic acid. But we thought it would be important to do a head-to-head comparison just to get an idea of the activity of nucleic acids and their ability to trigger the contact pathway. We also thought that most of the methods that are used to purify DNA or RNA would also co-purify polyphosphate. So if you ground up a cell that also had polyphosphate in it, you're likely to purify the DNA plus whatever polyphosphate they had. And then if you did clotting assays, maybe the activity you were measuring was a combination of the two. So we looked at the methods that were published for isolating DNA to do these kind of assays. And almost everyone uses these QIAGEN kits as a convenient way to purify DNA. And actually, Thomas Rene's lab has started using these QIAGEN kits to purify polyphosphate from cells. They're quite effective at doing that. So we did that, and we were really surprised by the results we got. So this next few slides is sort of a cautionary tale. If we purify DNA from HEK293 cells or human neutrophil nets, it had measurable procoagulant activity, but it wasn't particularly active. But if we purified those same DNAs using QIAGEN kits, it was way more procoagulant. Now, how do these QIAGEN kits work? Well, they have what they call a silica membrane. And silica has these negatively charged groups on them. And in the presence of a really high concentration of sodium ions, so typically they have you make the lysate in sodium iodide, sodium bridges between the silica these silicic acid moieties and negatively charged polymers like DNA or polyphosphate. And you can wash away everything else, and then you simply add water to the, you wash the membrane with water, and the sodium ions come off and the DNA is repelled from the silica, and so that's the basis by which these work. Now the unfortunate aspect of that is that silica is incredibly procoagulant. There are APTT reagents on the market that are made of silica, colloidal silica. It's actually one of the favorite things to make an APTT reagent about. So what we found was that if you just did a mock purification and took the water that came out of one of these QIAGEN columns, it actually has a lot of silica particles, and they will shorten the clotting time of plasma quite effectively. And what we found was that if we took short-chain polyphosphate or a relatively short DNA like lambda DNA, which has very little activity, and we repurified those on a QIAGEN column, they gained a lot of apparent procoagulant activity. I won't go through all the experiments, but we did controls to show that basically if you use one of those QIAGEN kits to isolate DNA or polyphosphate, you risk contaminating the material with highly procoagulant silica particles. Now, isolating DNA using, say, phenylchloroform does give you a procoagulant material and that procoagulant activity goes away with DNase-1. So we certainly confirmed that nucleic acids are indeed procoagulant, but the cautionary tale is don't use QIAGEN kits if you're going to do activity assays. Now, this is a rather complicated set of experiments, and I'm just going to make one point here, but this is a paper that Josh Gasowitz in my lab, the graduate student in my lab, published earlier this year. And what he did was he looked at the ability of polyphosphate versus RNA to enhance all the various reactions that go into triggering the contact pathway. So there's actually 7 reactions that are involved. There's reciprocal activation of prekallikrein and factor 12, as well as autoactivation of all of these molecules, and then 12A activating 11, as well as the back activation by thrombin, et cetera. So there are 7 reactions. And what we discovered was that DNA or RNA that we isolate from cells, although it does have measurable activity, is really low, and it was hard to do any biochemistry with that. But what we discovered was that the synthetic RNA, poly-G and poly-I, are way more procoagulant. And this is consistent with a paper from Preissner's group that said that hairpin RNAs and G quadruplex RNAs and DNAs were more procoagulant. And poly-G and poly-I fold up into G quadruplex structures quite readily. So these were very active and supported most of the reactions. But the point I want to make here is that polyphosphate was good at supporting all 7 of these reactions. That's the pluses here. The missing link, sort of the weak link, was this reaction and this reaction, factor 5 and factor 6. So nucleic acids are actually quite poor at activating 11, either through autoactivation or linking it to 12A. And what this, to my mind, raised is the possibility that nucleic acids could be supporting this aspect of the contact pathway, leading to bradykinin generation and the pro-inflammatory aspect, without necessarily linking it very well to the formation of a fibrin clot. So it's a potential cofactor for generating kallikrein activity, cleaving bradykinin out of high molecular weight kininogen, without propagating that whole set of reactions down to the generation of fibrin, whereas long-chain polyphosphate will do all those. So I think it bears some real re-examination as to what nucleic acids might be doing to subsets of the reaction of the contact pathway. And then finally, I just wanted to mention that we set up a really interesting collaboration with Ed Conway, and basically we provided reagents and expertise on polyphosphate, and he ran away with it to do some fascinating studies on the complement system. And so far, a couple of papers have come out on that. What we found is that polyphosphate really is a potent inhibitor of the assembly of the membrane attack complex of the complement system. And it's also an outstanding cofactor for C1 inhibitor inhibiting C1s. And interestingly, C1 inhibitor is the major inhibitor of the contact pathway. Polyphosphate does not accelerate that inhibition. So polyphosphate maintains its procoagulant activity by not being a good C1 inhibitor cofactor for going after the contact pathway enzymes, but being a really good cofactor for downregulating the complement system. Now, why polyphosphate is important in upregulating clotting and downregulating complement still, I think, remains to be understood. So I'd like to just summarize by saying that microbial polyphosphate, long-chain polyphosphate, and I'll add there that Kornberg's lab published an interesting survey of rat tissues in which he reported that brain, for example, contains long-chain polyphosphate, hundreds of phosphates long. So there are potential endogenous sources of long-chain polyphosphate that could be triggering clotting following injuries, for example, traumatic brain injury. So microbial polyphosphate and potentially long-chain endogenous polyphosphate is a really good trigger of the contact pathway. Platelet-sized polyphosphate, we think, has that really narrow size distribution because its job is to accelerate downstream reactions and leave the contact pathway alone. Polyphosphate is an important regulator of complement. We think it's a target for novel antithrombotic drugs, and I think it's very safe to say that we've probably just scratched the surface and that polyphosphate almost certainly is doing things other than just modulating the clotting and complement cascades. With that, I'll thank you for your attention and be happy to take questions.
Jim, I'm wondering whether platelets or megakaryocytes can take up polyphosphate and maybe bacteria in the gut as a source.
Yeah, so, we don't know. And We've done some experiments with cultured megakaryocyte-like cell lines, and they certainly accumulate polyphosphate. So I think the megs are making polyphosphate that gets put into the dense granules. But whether they can take up exogenous polyphosphate, we simply don't know. There are many still unanswered questions. We don't know if the polyphosphate that's in the platelet dense granules turns over at all. That's on our to-do list. You know, are they born with all the polyphosphate they're going to make, or are they constantly making it and degrading it? There's a lot of simple things that we haven't just had a time to look at. Yeah.
Polyphosphate must have a whole bunch of other biochemical properties. I know there's polyphosphate in every nuclei, for instance.
Right.
And it's a very powerful inhibitor of tyrosine phosphatases.
Yeah.
Do you know some of those reactions, why it's present in every nuclei?
Yeah, we don't know. It certainly will interact with very positively charged proteins like histones and HMGB1. We've only studied that once they get outside the cell. Histones are procoagulant. Polyphosphate is procoagulant. The combination of histones and polyphosphate is way more procoagulant. My colleague Ray Rosé has shown that when HMGB1 binds polyphosphate, it becomes a much better activator of endothelial cells. So I think there are many binding partners for polyphosphate, and as you're hinting at, it probably does many other things, and but we mostly don't know. You must, yeah.
Have you looked at platelets with dense granule defects?
We have. So we've looked at Hermansky-Pudlak syndrome platelets, and they lack the ability to antagonize TFPI function, the releasates, and you can restore that by adding polyphosphate to them. The other thing that I'm aware of is that there were some colleagues in India, and I'm blanking on their names now, who published an interesting paper in Blood a few years ago, in which they knocked out the enzyme, one of the enzymes that converts inositol hexakis phosphate into inositol IP7 or IP8, basically. So inositol hexakis phosphate is an inositol ring with a phosphate on every OH group. And this enzyme puts another phosphate on to make a pyrophosphate on one of those OH groups. In yeast, if you knock out that enzyme, you drastically downregulate polyphosphate accumulation in the yeast vacuole. So there's some connection between IP7 and polyphosphate synthesis. So they they kind of wondered if that was true in mammals as well. And they knocked out, there's two isozymes for this enzyme in mammals. So they knocked out the one that was made in megs. And what they found is that the level of polyphosphate that was packaged into the platelets, in the mouse platelets, went way down. And that those mice had a mild bleeding problem and were protected against thrombosis. So there is evidence that, that it's important.
More questions?
Thanks, Jim. That was a fantastic talk.
Thank you.
Do you know what the half-life is of platelet polyphosphates, and are there endogenous phosphatases that degrade it?
Right. So the half-life, it's a really interesting question. If you simply spike polyphosphate into blood or plasma or serum, it will decay with roughly a 90-minute half-life. And I have a manuscript sitting on my desk that I just need to get submitted, darn it. But I had a student who took that on as a project, and I thought, well, okay, there's phosphatases in blood, they're doing this. And so he took serum, actually, it was the easiest thing to look at. He put every phosphatase inhibitor that you could buy. The only one that did anything was EDTA. And so in desperation, he boiled the serum for 20 minutes and almost everything precipitated, and he took the supernate and cooled it back down, and 90-minute half-life, right? So long story short, it's the combination of the slightly alkaline pH of blood and millimolar amounts of calcium causes chemical decay of polyphosphate. So it's dominated by this sort of chemical decay. I think it explains why every compartment in biology in which calcium and polyphosphate are stored together is acidified. It's the only way to stop that chemical hydrolysis, basically. And it could explain why a lot of subcellular compartments are acidified. But it does mean there's sort of like a built-in decay of polyphosphate once it gets out of the cell.
So Jim, And do you think that polyphosphate is coating cells, though? Because Thomas thinks that they're coating platelets and endothelial cells after it's released, to some extent.
Oh, for sure will bind to cell surfaces. What Thomas Rene is saying is that when the polyphosphate is first released, that it's packaged with, really densely with all these metals, and it takes some time for that to dissolve. Almost certainly true. How long that takes is not so clear to me. Certainly once you release it, the concentrations of the metals in polyphosphate are they're well below the solubility limit. So if you take 1.2 millimolar free calcium, you can easily put micromolar concentrations of even really long chain polyphosphate in. They're water soluble. So, but there may be some time delay, where these things are dissolving once they're released.
Jim, thank you so
Okay,
much.
you're welcome.
The next three speakers that we have are some of our excellent PhD students who will give talks, whose posters are also out in the hallway. So I encourage you to go and look at all of the posters. They, I hope, will write on their CV that they followed with a talk from really esteemed speakers, including a Nobel laureate. So make sure that goes on your CV somewhere. I had no decision-making in who speaks, and the first speaker is Brian Lynn from the Prysdale Lab.
Thank you, Ed, for that short introduction and the opportunity to speak at this symposium. Before I begin, I want to do a small disclaimer in that these talks are supposed to be like 7 minutes long, but I want to say that I had no hand in there being not a dedicated timekeeper, and I see that Ed has his timer up, but he's timekeeping. So today I'll be showing you work I've been doing on how some human viruses affect hemostatic abnormalities. And this is based on envelope viruses, where some viruses that our lab has shown can derive an outer membrane structure known as the envelope from the host, along with the main initiators of coagulation, tissue factor and anion phospholipid, with our hypothesis being that any virus that can derive this envelope with these coagulation factors can induce or cause hemostatic abnormalities. To demonstrate this, we used two very distinct families of viruses, with the representatives being herpes simplex virus and dengue virus. Both of these viruses have been known to cause hemostatic abnormalities, and there are also huge burdens to the healthcare system worldwide. Herpes simplex virus is the major cause of cold sores, genital herpes, infectious blindness, as well as viral encephalitis, whereas dengue virus can cause breakbone fever or the more severe forms of hemorrhagic fever and shock syndrome. Our lab has shown that tissue factor, as well as anion phospholipid, are incorporated into several members of the herpes viridae family, and that this allows for factor 7A to bind and cleave factor 10 to factor 10A. That ultimately results in clotting, and if that goes out of hand, it can result in pathologies such as thrombosis. These clotting proteases are also involved in cell signaling, leading to inflammation. And as our lab has shown, the virus can make use of this clotting protease activated cell signaling to enhance infection in vitro as well as in vivo. I have used different sized gold beads to label different antigens simultaneously on the virus surface. Here I've been using the virus-specific or herpes simplex virus-specific marker, glycoprotein C. We also have anion phospholipid and tissue factor. And this allowed us to confirm that a single virus particle has the main initiators of coagulation on its surface. However, tissue factor activity, as James Morrissey said, talked just earlier, its activity isn't normally exposed to the blood. So we tested for the tissue factor activity by looking at clotting in pooled normal human plasma, where a lower clotting time means a faster clot is formed. We used viruses that were grown in sister cells that either express tissue factor or do not express tissue factor. And what we found were viruses that have tissue factor require 100-fold less virus in order to induce clotting. We moved on to our next envelope virus, dengue virus. And similarly, we found that dengue virus also incorporates tissue factor as well as analog phospholipid. Seen here are two distinct virus particles where they are labeled with the dengue virus specific E protein. We confirmed tissue factor function by including dengue virus. There were different serotypes of dengue virus, and this is work done by Michael Sullivan in our lab, that were grown in cells that express tissue factor, and we found that adding them into plasma would induce clotting, suggesting tissue factor was functional. Now, in addition to tissue factor, viruses have their own, or they encode their own arsenal of surface proteins. Herpes simplex virus, in particular, has glycoprotein C. Glycoprotein C, or GC for short, is a very important virus protein, as is important in infection, as it mediates cell and virus interaction, as well as host immune response escape. Furthermore, it has been shown that GC can bind to factor 10, which led us to explore its ability to regulate factor 10 function. And we saw that when we added viruses that had GC, they had a greater procoagulant activity compared to GC deficient virus. And this could be explained by following factor 10 activation in purified protein and virus chromogenic assays, where viruses that lack tissue factor have very little to no factor 10 activation, but in the presence of tissue factor and GC, we see enhancement by GC in factor 10 activation. Interestingly, when we added a soluble form of GC, we could enhance purified tissue factor mediated factor 10 activation, further fueling our curiosity as to how this virus protein has evolved to affect clotting. Previous work done by James Morrissey's lab, where he used nickel-ketone lipid to orient by tethering a histidine-tagged soluble form of tissue factor to restore its biological function. And we're using this similar method to study the function of soluble GC with a histidine tag. When we followed factor 10 activation, we saw that without nickel-ketone lipids, we see very little or no factor 10 activation. But with the inclusion of nickel-ketone lipids, soluble tissue factor, as expected, had a restored biological function in enhancing factor 10 activation through factor 7A. And interestingly, we saw that soluble GC also enhanced in the absence of tissue factor. And when we add the two together, they both simultaneously enhance factor 10 activation. To further understand how soluble GC mimics tissue factor function, we looked at binding assays, and we used microscale thermophoresis to study this, and this technique revolves around the differential movement of molecules across a temperature gradient. What we found was that the apparent binding of factor 7A to soluble tissue factor was decreased when we added soluble GC. And this suggests that soluble GC is pulling the factor 7A away from the tissue factor. Overall, I have shown that two distinct families of viruses derive functional tissue factor and analog phospholipid, and that a herpes virus protein, glycoprotein C, can enhance plasma clotting as well as mimic tissue factor cofactor function in enhancing factor 7A activity. However, this requires membrane association. If you have any more questions or you want to talk more, you can see me at poster number 13. Thank you.
Our next student speaker is Beverly Bakir from the Hancock Lab.
Great. Hi there. So my name is Beverly Bakir, and I'm a PhD student in Dr. Hancock's lab. And the focus of my project is cellular reprogramming during sepsis pathogenesis. So though we've had a couple of definitions of sepsis, the definition that I typically use, which is pretty general, is the host immune response to infection that's dysregulated that can lead to organ failure and death. There are 26 million cases of sepsis annually and has a high mortality rate of 30%. Even those that survive sepsis have poor long-term outcomes. And this is in part due to the clinical dogma that initially was that was initially used to describe sepsis. Initially, it was described as having an early hyperinflammatory phenotype, followed by a subsequent late immunosuppressive phenotype. But as we've done further research, there's been a need for updated clinical definitions, which was done last year. And this basically tried to encompass our updated understanding that that phenotype is actually happening rather concurrently rather than in a subsequent fashion. Now, even with our updated understanding, as well as awareness campaigns such as World Sepsis Day on September 13th, there's still a lack of effective therapies. And this is in part due to our need for further understanding of the pathogenesis of sepsis, as well as to further address the complexities of this disease. So the way I'm planning on addressing this complexity is to try to look at the lesser studied characterization of cellular reprogramming. Now, cellular reprogramming is a cell's inability to respond to a secondary stimulus, which results in cellular or immune amnesia. Monocytes taken from sepsis patients actually exhibit this inability to respond to agonists, so therefore links it to our cellular reprogramming phenotype. Additionally, our lab has previously generated a 31 gene cellular reprogramming signature to be able to, which has been linked to be able to predict whether or not a patient will go on to develop sepsis. So with these ideas in mind, my hypothesis is that during early sepsis, reprogrammed monocytes can cause a dysregulation in the host immune response. So my three main aims are to verify that monocytes are actually able to drive this phenotype. Secondly is to characterize these reprogrammed cells. And lastly, it's to define the inflection point that leads to cellular reprogramming. So though the link between cellular reprogramming and sepsis is still not fully understood, we have been able to link a cellular reprogramming gene signature with the development of sepsis. And what I did here was I used CyTOF, which is a combination of mass spec and flow cytometry, and I used lysed whole blood and bound them to antibodies that were conjugated to heavy metal isotopes. This allows me, using this technology allows the identification of a number of different parameters, as many as 40 parameters for each cell. So basically, as a cell gets atomized into the machine, a time-of-flight detector is able to identify a number of different parameters per cell. So I used this methodology in taking first my 3 different conditions. So I had my vehicle control as just my basic control, but I was really trying to compare my inflammatory or single lipopolysaccharide or LPS treatment versus my double LPS, which was also my reprogrammed phenotype. So basically, I wanted to use this methodology and used my 17 marker antibody panel with these particular conditions. And my 17 marker antibody panel basically takes 8 of my antibodies and basically identifies a specific cell subset within the whole blood that I'm interrogating. And once I am able to identify that particular cell subset, I'm actually able to track the expression of my cellular reprogramming markers as well as my inflammatory markers. So basically, this particular graph shows that when I have a single LPS stimulation, I'm seeing increased expression of my inflammatory markers, which is what I'm basically expecting. Now, when I look at my reprogrammed marker expression for my double LPS group found here, you're seeing we see 3 out of the 5 of these particular markers being upregulated. So though not perfect, there seems to be a link with this particular subset of cells and the expression of these cellular reprogramming markers. So my next step was to basically characterize cellular reprogramming. And so I initially did this with murine embryonic stem cell derived macrophages. So you basically take embryonic stem cells through a series of steps and generate these mature macrophages. And I also used BMDM's bone marrow derived macrophages with the same background, murine background as a comparison. But ultimately what I did was I basically put them into the same three conditions that I noted earlier with expanded time points. These expanded time points I'll elaborate on in the next slide. But ultimately what I did was I took these cells, isolated RNA, and then sent them out for RNA-seq. From this, I was able to look at the murine homologs that were found in the differentially expressed genes that were found in the double LPS stimulation. And I can see that of the murine homologs that I found of my gene signature, I see increased expression, fold change expression of my double LPS compared to my single LPS. Ultimately, I'd like to be able to use this methodology to make macrophages from human-induced pluripotent stem cells. And we've prioritized 3 genes from our cellular reprogramming signature to be knocked out. And so therefore, our plan here is to generate macrophages from these mutants and then reprogram them with specific agonists so that we can be able to determine the functionality of these particular genes in the development of cellular reprogramming. So as I noted earlier, I've expanded my time points but still had the same 3 conditions. And when I looked at this heat map, when I plotted out the heat map, you can see that the clustering is mostly driven by condition rather than time. Looked at another way, on a PCA plot, you can see pretty distinct clustering of vehicle control as well as their double LPS group. But when you look at the single LPS, it looks like time seems to be playing a larger factor here. But what's really interesting is that when you look at in the double LPS group, 2 hours after the secondary stimulation of LPS seems to be enough to drive this cellular reprogramming phenotype. So it looks like I should be interrogating earlier time points for this inflection point to be occurring. So I hope I've been able to show you that reprogrammed whole blood has increased expression of our cellular reprogramming signature, and that ESDMs are able to mimic these reprogrammed macrophages, and these murine homologs do exhibit this gene signature. I'll be doing applying this methodology to using human-induced pluripotent stem cells and to make them into macrophages, and also that the inflection point for reprogram is likely earlier than I initially expected. Thank you.
Our third of 3 speakers is James Bayless from the Castrop Lab, and you've met him already.
James originally had 56 slides, but we've cut him down to 7. So I hope you're going to have to manage.
Hi. Thank you, Ed. Thank you, Ed. Thank you, non-Ed organizers. I'm still James. I still work for Christian. He has not fired me today. And this is self-propelling particles loaded with tranexamic acid stop bleeding following trauma. Bleeding kills. Everyone bleeds. Everyone dies. Therefore, there's a big group of people who bleed and then they die. This is especially true in trauma, in which bleeding is the leading cause of preventable death. Bleeding is responsible for 1 in 3 in-hospital trauma deaths, and if not managed quickly, can progress to hyperfibrinolysis and coagulopathy. Another situation in which bleeding is a big problem, but fewer people die, is during surgery, in which it consists of a large clinical burden. Bleeding is responsible for infection, procedural error and revisions, increased OR time, and increased requirements for blood transfusions. There are many agents which are used to stop bleeding, which can act as stimuli to the coagulation cascade, which we all know and love. However, these are often fail to stop bleeding. One reason for this is transport. Transport limitations can complicate the management of bleeding. This is true for topical agents and sealants, in which high pressures can expel the agents. It can be difficult to apply in all the nooks and crannies of wounds. Number 3, sometimes you literally cannot see what you're doing. And 4, it often requires adjunct compression to slow blood flow. Similarly, intravenous hemostatics also face transport limitations in that sometimes you have to give very high doses systemically to reach adequate doses at the wound site. Hypothesis: propelling hemostatic agents will improve their ability to stop bleeding. So here you have a damaged vessel. It's bleeding out. It's a bloody mess. You apply the particles loaded with a hemostatic agent. Maybe you're close, maybe you're not, but it doesn't matter because these particles can propel all over the wound site, including to the site of damaged vasculature, to stop the bleed. We did this using calcium carbonate microparticles. These particles propel and improve the dispersal of hemostatic agents into bleeding wounds. Shown schematically here, we have calcium carbonate particles in green, mixed solid via solid-solid mixing with an organic acid. When you place these in any aqueous solution, including blood, the acid dissolves and the calcium carbonate reacts, producing carbon dioxide and propelling in all directions. This mixture can also be loaded with cargo, hemostatic cargoes. We've tested multiple things, and the propulsion improves the dispersal of these agents. Seen in the bottom here, this is histology of mouse livers on the left, in which we applied propelling particles. We see a higher level of deposition of fluorescent nanoparticles compared to applying the same formulation which does not propel. Using this formulation, we impregnated it upon gauze and used thrombin as the hemostatic agent which we are propelling, as well included as the organic acid, tranexamic acid, which is a clinically used antifibrinolytic agent. On the left here, a photo of the gauze. One part has calcium carbonate and thrombin. The other part has tranexamic acid hydrochloride as the proton source. In this swine femoral artery injury model, femoral arteries were accessed and punctured, and the wounds were packed with propelled thrombin gauze. Again, a similar formulation which does not propel, or combat gauze, which is a clinical standard for this type of bleeding. We found that propelled thrombin significantly increased survival compared to the other two groups after 3 hours. Using a similar formulation, we showed that the same propelled thrombin plus TXA gauze reduces surgical bleeding in sheep. First, we tested in an endoscopic surgical model of bleeding in which the nasal turbinates of sheep were accessed endoscopically, and we took a nice big bite out of the tissue to start bleeding. Gauze was applied with forceps, either propelled thrombin and TXA gauze or plain gauze, and it was found that propelled thrombin and TXA gauze significantly reduced bleeding. More excitingly, we did a carotid artery puncture in these sheep. Carotid arteries were accessed, and we stabbed a hole in them and packed them with either propelled thrombin and TXA gauze or flow seal, which again is a clinical standard for this type of bleeding. When the dressing was left on the wound, we found that 100% of sheep achieved hemostasis after receiving propelled thrombin and TXA gauze, compared to only 25% with flow seal. However, if you tear off that last gauze, they both perform similarly, with only 25% of sheep achieving hemostasis. Next, we tested a formulation which contained only TXA and no thrombin, and we found that propelled TXA was also able to stop bleeding following trauma. In a mouse tail clip model, self-propelling TXA was applied as a powder. After tail clip, bleeding was quantified, and we found that self-propelling TXA significantly reduced blood loss to 2 doses of IV TXA, and again comparing to a topical TXA powder which did not propel. Propelled TXA gauze, which again was 1 gauze with calcium carbonate, 1 gauze with tranexamic acid, was again packed into our pig femoral artery injury model. Survival was measured over 3 hours, and in this group, 67% of pigs survived, which looks higher than the other two groups. However, the difference was not statistically significant. Interestingly, 1 hour after application of propelled tranexamic acid gauze, the concentration of TXA in the plasma of these pigs reached therapeutic levels by 1 hour, and this poses an interesting opportunity to reduce the time to treatment using tranexamic acid in trauma. In conclusion, self-propelling particles can increase the efficacy of existing hemostatic agents. Calcium carbonate particles improve the dispersal of these agents. Propelling thrombin and TXA was useful for stopping massive traumatic and intraoperative bleeding. And propelling TXA is also useful for stopping massive traumatic bleeding. Next steps were developing a propelled thrombin formulation for endoscopic gastrointestinal bleeds, as well as a propelled TPA for treating deep vein thrombosis. Here's a picture of Castrop Lab. They're sitting over there. Their projects are cooler than mine. I encourage you to go talk to them too. Thank you.
I'll call her Agnes Lee. Dr. Lee is a hematologist, professor of medicine here at UBC, and she's going to introduce our next speakers. Are they here? No, come on up. Are they here? I hope so. Holly, Sheila, they're here. That's a good thing. Hang on one sec. One sec. Okay. All right. Whoops, that's not good. Hang on. Here we go.
Okay.
And it goes right on.
Thank you very much, Ed. Sorry, I sort of put myself in the agenda there. But thank you for inviting me to actually introduce our next patient speakers. But before I introduce them, I just want to give you a little bit of background to put in context the challenges that they face. And before you leave here thinking that only bleeding kills, after hearing from James, clotting unfortunately also kills. So I think many of you in the audience are very familiar with the most common clotting problems that we face, including MI, myocardial infarction or heart attacks, strokes, pulmonary embolism, which is blood clots in the lungs, as well as deep vein thrombosis. And together they are the most common clotting problems. They are also the leading causes of death globally, and in the US, every 5 minutes somebody dies of a heart attack. Every 4 minutes, somebody dies of a stroke. And for patients who have pulmonary embolism or DVT, or venous thromboembolism, 30% will die if they're left untreated. And together, they're responsible for 17.5 million deaths per year. And unfortunately, this is only going to go up. So we thank you very much, all of those who are working on new therapeutics to help with treating patients who have clotting problems. And sadly, thrombosis is also the #1 killer of women worldwide, and this far exceeds all cancers combined. So for Canada, our numbers are obviously smaller, but it still occurs in about 200 Canadians per year, and up to about 10,000 Canadians die from PE each year. This is an underestimate because that we know from autopsy studies, for example, 80% of blood clots in the lungs are found at autopsy were not diagnosed before death. So we're not doing a good job picking up this early on. We know that PE alone causes more deaths per year than AIDS, breast cancer, and highway fatalities combined. So big problem. And unfortunately for survivors, a third of them will have another clot in the next 10 years, and a third of them will be left with chronic complications. And so I hope to put all this in context, and now I want to introduce you to the two patient speakers. Sheila and her daughter Holly are both my patients, and I'm very thankful to them for coming to share their experience and their inspirational stories about their challenges with living with thrombosis.
So as you can all see, I have emotional problems talking about my stroke. So bear with me, I'll try to make some jokes and lighten it up, but my name is Holly Walker, and what I wanted to let you all know is the positive side to the story, is that this past spring, I had the opportunity to be assistant ski guide on Mount Logan expedition in the Yukon Territory. On May 23rd, 2017, we reached the summit at 5,959 meters, Canada's highest peak. I thought it was a bit funny that there was a photo of a summit and just on a second go on the slide. Anyway, Mount Logan has low ascent odds due to extreme weather, and we persevered through poor visibility and high winds and low temperatures down to negative 40 degrees Celsius. Here's me on the summit of Canada. So just showing you that you can start at the bottom and get to the top really easy. Flashback to January 11th, 2009. I was 28 years old. I suffered from a headache and I did not feel well on a ski trip in Colorado. I awoke early on January 15th. I was unable to get out of bed and use the bathroom. I was incapacitated and driven to the hospital in Durango by a friend. Hospital staff believed that I was a youth that had been partying too hard on drugs. They had to be convinced to treat me. An ER doctor performed a CT scan on my brain and diagnosed that I had an inoperable tumor. I was then airlifted by air ambulance to a neurological ward in Denver. My parents flew south from Vancouver and pressed doctors to treat me further, not believing the first diagnosis. Nearly 6 days after initial symptoms of that first headache, I was diagnosed that I had had a stroke, an extensive cerebral venous thrombosis and associated left thalamic infarction. Excuse me if I was wrong with that terminology. Steroids were used to decrease my swelling and my condition improved. I would survive. Today I want to thank Vancouver General Hospital, where I was airlifted to and treated further. I want to thank GF Strong Rehabilitational Center, who trained me for 3 months to reactivate my brain and the function of the right side of my body. It would be a long road to recovery. Physical therapy, occupational therapy, speech and language therapy, emotional therapy. As you can see, I'm still working on it. When I first arrived to GF Strong, I cried alone by myself, sitting on my new bed, and made a goal to recover and to be able to function than ever before. I knew that I stood a chance when I beat my physical therapist at a game of ping pong with my right hand. In early February 2012, just 3 years after my stroke, I aimed for the top. So there's a bit more of... I aimed for the top and to summit North America's highest peak, Denali, at 6,190 meters. I trained hard that winter, and I had conflicts from several doctors that high altitude and thick blood was a bad idea. I communicated with Dr. Hackett, a high altitude specialist, and he told me that were far greater risks: acute mountain sickness, HAPE, haze, frostbite, avalanches, and other objective hazards to worry about than my less than 1% chance of suffering from another stroke. He recommended that I stay well hydrated and avoided being tent bound. This is me at high camp. On the Denali expedition, our high point was only 5,400 meters, just above Camp 4, due to poor weather at altitude. No headaches or further complications gave me the confidence to try new challenges and to continue living my life to the fullest. I'm now an ACMG apprentice ski guide working in British Columbia's backcountry, and I continue to ski and climb around the world. To learn more about past and future expeditions, feel free to check out my website, hollywalker.ca. My message to the outsiders are very simple. Just try. You only live once. And hell, you might just reach your summit. Now I want to introduce you to my mother, a member of my clot club, who was a great supporter in treating me with my rehab.
Okay, so what you need to know about our family is that I've had 3 deep vein thromboses from 2013 to 2016. No matter what medication I was given or anticoagulants, I kept clotting. And it wasn't until Dr. Lee decided to test me for several different tests that the factor 8 came back, that I have 3 times the level of factor 8 than a normal person. So I went and saw Ed Prysdale, who took our blood, Holly's blood and my blood, and it's supercharged. It's not that I have too much, it works too hard. So now they're looking at the genetic factor. Because my son, 2 years ago, had a pulmonary embolism. And the size of clot in his lung was the size of a baseball. And he had extensive damage to his lungs. So we are very pleased and very thankful to Dr. Lee, Ted Prysdale, and Ed Conway, because now they're doing research on our family to determine what is wrong with our blood. Because definitely we do have a family clot problem. So I want to thank you all very, very much. It has been a very emotional, long ride since 2006, or 9, I should say, 9. But I also should mention in closing, my mother, at the age of 46, in 1967, had a pulmonary embolism. And when Holly had her episode, no one would listen to me, that there had to be a connection. So we've all had clotting problems, and now they know there's a connection. And my husband, who's in the room, always loves to tell people he is associated to the Clot Club through marriage. Thank you. Any questions?
I forget who's speaking next. Oh, yeah. It was like pulling teeth for me to get the next guy to talk. He's really modest. That's why. And I'm excited to introduce the CBR director to finally give a talk at one of these beautiful events. Ed has an amazing education history that started with an electrical engineering degree, and then he jumped disciplines to medicine, both at the University of Toronto. Following Toronto, while on a rather extended sabbatical, 14 years, he did his PhD and his MBA, both in Belgium, Leuven. In addition to his tireless work at the helm of the CBR, I'm sure with the help of bottomless cups of coffee, Ed somehow finds time to be involved in considerable charitable work, especially in Uganda, but not exclusively in Uganda. And he's even relearning how to play classical guitar, like you. Ed C., we all thank you for orchestrating this great symposium. I'm emotional too. And among all the related responsibilities, agreeing to give this infernal talk. Thanks a lot, Ed.
Thank you, Ed. So this is the group who, just so I won't forget to do it, and it's really a little lost. But this is the team, and I'm sorry that I buried some of them there. The work. This is the group. This is Tara and Hura. Some of them are here. Victor, Nushin. Back there, hidden is Lana, but you've seen her at the poster, and she gave a short talk. This is Piyush is right here, and Jenny is right there, and David is hidden back there. Most of this work that I'm going to describe was done by Tara Fernandez, who's a postdoc who's actually left to biotech, and Piyush, who's actively working on the projects. So obesity, it's common. 30% of Western world is afflicted, and it's on the rise in the developing world, and it comes with all kinds of problems. It comes with, here's a very obese mouse with, or maybe it's a rat, with a retinal problems, a retinopathy, diabetes undoubtedly, but it suffers from a chronic inflammatory disorder with endothelial dysfunction, as well as vascular disorders, including atherosclerosis, hypertension, and a higher risk of venous thrombosis. And it's not really a surprise, considering that adipose surrounds all our vessels, essentially, and clearly must crosstalk then with underlying smooth muscle cells and vascular endothelial cells when dysfunctional, causing disease. Now, several years ago, we came across a molecule known as endothelin. It was just first called endothelin because it was believed to be endothelial in origin. In fact, it's not. Excuse me. And now I refer to it as CD248. It has low expression in most normal adult tissues, highly upregulated in inflamed tissues, mostly mesenchymal cell origins, expressed in smooth muscle cells and parasites, and particularly elevated in adipocytes. Now, what about adipose? Now, in very simple terms, there are effectively two types of adipose. There's white adipose, which is what's right there, okay, which is highly associated with cardiovascular disease, and it stores lipids. And then there's the good adipose, which is so-called brown adipose, which essentially burns off lipids to generate heat. And that's the good stuff. So what do we want to do from a therapeutic point of view, other than to eat less and eat good foods, is to change this abdomen, because that's where it's associated with cardiovascular disease and thrombosis, and transfer it into this sort of thing where you have more brown adipose tissue, less white adipose tissue. Now, this can be done with cold. Actually, people sell cold belts where you can wear them for 18 hours a day, not particularly comfortable, or catecholamines. Not okay, I got that. All right. But then there's a search for wonder drugs to do that. Now, CD248, I said it's expressed by adipocytes. It's not expressed in brown adipose tissue, but it is highly expressed in white adipose tissue. So what's it all mean? And in fact, in humans, this is in collaboration with a group in Karolinska where they studied humans, obese and non-obese people. If you took subcutaneous adipose tissue from these people, donors, essentially, and measured their RNA levels of CD248, there was a significant correlation, so a direct correlation of CD248 levels in the obese versus the non-obese. That also turned into an excellent predictive marker for insulin resistance. So these are insulin resistant obese, these are insulin sensitive obese. So again, CD248 is significantly elevated in those individuals. And it also translates into patients who are lean with type 2 diabetes versus healthy matched controls, that CD248 is significantly elevated. In fact, this marker is better than any marker that's ever been described in terms of composites of other signs. In addition, if you take the white adipose tissue from these individuals and you incubate them in a media for a few hours, they secrete cytokines, inflammatory cytokines, and the levels of CCL2 or IL-6 are directly correlated with the amount of CD248 in those fat tissues. So they're pro-inflammatory as well, or CD248 is. So to work out the mechanisms and what is CD248 doing in vivo, we generated mice that lack CD248. These mice are perfectly healthy looking and fertile. We put them on a high-fat diet, so this is weight over time. And you can see they increase weight. This is the wild-type mouse. This is the knockout mouse. They gain significantly less weight over that time period. These are their white adipose tissue in the knockouts, so you can see that these are significantly smaller than in the wild-type mouse, and that's shown here. So the white adipose tissue is decreased, which is what you want. And in fact, the brown adipose tissue is increased in weight and size. So you can see that here. So they're doing exactly what you want. In addition, actually, the livers are significantly different. This is the wild-type mouse at the end of the high-fat diet, so it doesn't take a hematologist or rather a gastroenterologist to see that this liver is fatty. But this is a normal liver, and that's the knockout mouse after that high-fat diet. So it looks entirely normal. Glucose tolerance tests. These mice are protected. If they are the knockout mice are protected against glucose intolerance, and they have improved insulin tolerance as well. So they're not diabetic. All right, so CD248 then is associated with increased white adipose tissue, with obesity, with insulin resistance, and diabetes. But what's the link to thrombosis? So Piyush has been doing these studies. These are very preliminary, but some but very convincing at the same time. So he's used a couple of models, but I just described the stenosis model. So this is in mice, knockout mice. These are not on high-fat diet. These are just knockout versus wild-type mice. And then this is a 90% ligation at just below the renal arteries, renal vessels here, and you leave these mice for 24 hours or 48 hours, and then measure the size and the weight of the clot. So these are the clots. You can see this is the wild-type clot. This is a knockout clot, significantly smaller. These are in females. You can see the difference in weight in the wild-type, but in the knockout they're significantly smaller. Indeed, several of them do not develop a clot at all. That never happened in the wild-type mice. In males, it's the same picture, except males tend to have larger clots. So again, it's a significant difference. The knockout have smaller clots. And that's true at 48 hours as well. When you take the blood, whole blood from these mice, the wild-type mice and the knockout mice, and you perform ROTEM, or equivalent would be TEG, but it measures clotting parameters, the clotting time is significantly prolonged in the knockout mice as compared to the wild-type mice. The rate of clot formation is significantly slower in the knockout mice as compared to the wild-type mice. So we raised the question, although we don't necessarily have the, from these studies we really haven't identified the pathway. What we thought that tissue factor was a likely candidate. There were other reasons why I'll go through later. But we asked, could CD248 be interacting with tissue factor to enhance the generation of factor 10A, and then in so doing promote clot formation? So first we just showed by co-localization studies, these are smooth muscle, these are primary vascular smooth muscle cells from the wild-type mice, from wild-type mice. And you can see CD248 expression here. This is tissue factor on the same cell, and the co-localization is here, and this is another cell. So we consistently revealed that CD248 co-localizes with tissue factor in these cells. There was some variability, but we could always see areas where it co-localized. So there were some differences cell to cell. So does this translate into any functional differences? And to test that out, first of all, we showed that the knockout down here, right at the bottom, knockout mice express tissue factor, if anything, a little more, which is a little curious, as compared to the wild-type mice, but they all express tissue factor. And we asked if we take these smooth muscle cells, culture them, and then look at the effect of CD248 on 7A activation of 10 in the presence of tissue factor, measuring 10A activation kinetically. So this is the rate of formation of, or generation of factor 10A in the wild-type smooth muscle cells, and that's the rate of generation of factor 10A on the knockout smooth muscle cells. So this is significantly different, although it does catch up afterward. So what we believe is CD248 is enhancing the procoagulant function of tissue factor, and that may have some role in explaining the effect in terms of thrombosis. So what's the link here then between obesity and thrombosis? I really haven't explained that, and I'm not sure I can. I can only speculate at this point. Now, to do that, we can go back a few years by papers by Samad and Wolfram Ruff. In the one that I'm referring to is in 2013, and they showed a direct interaction with beta integrin 1 with tissue factor, and when 7A was added to this mix, you activated PAR2, and PAR2 promoted a pro-inflammatory, an inflammatory response that manifested, at least in mice, they believed, in an obese phenotype. And if they knocked out PAR2, that was reversed. And they did some other studies blocking interaction between tissue factor and integrin to demonstrate this phenomenon. So we asked, okay, could this be the pathway by which CD248 performs. And we had some earlier evidence, because we had already established, which I haven't shown you, that CD248 actually directly interacts with integrin beta-1, which actually has an impact on adipogenesis. So this was the question: could CD248 interact with beta-integrin 1 and tissue factor, and in so doing promote activation of PAR2 to promote this kind of phenotype? I don't have the answer to that, okay? So I'm not going to show you any data. So in summary, CD248 is expressed by white adipose tissue, the levels of which correlate directly with obesity and with insulin resistance in mice and in humans. Deficiency of CD248 protects against high-fat diet-induced obesity, diabetes, and venous thrombosis in mouse models, and I would add also reduces atherosclerosis. CD248 interacts directly, or maybe not directly, but it interacts with tissue factor and enhances its procoagulant function, and reducing CD248 in whole blood reduces tissue factor-induced clotting and the procoagulant properties of tissue factor on the surface of vascular smooth muscle cells. And finally, CD248 interacts with tissue factor and with beta-1 integrin, and maybe that's a mechanism by which it regulates PAR2, and in so doing modulates the tendency to obesity. So finally then, CD248 we view as a potential new target for obesity and venous thrombosis. And at the same time, which I haven't discussed here, as we work out the interactions, the structural interactions, there may be roles for it in protecting against bleeding as well. Many contributors, I've already discussed our lab. There's a group in Laval who's been instrumental in helping us on the adipose angle, as well as locally, the Diabetes Research Group. That's Tim Kiefer, Michelle Kwan, Jim Johnson, Shuba Susie Lee, and then Ed and Brian have been helpful in the tissue factor work. And this is my group collaborators in the Karolinska, and finally I've got some collaborations with Morphotech, who've provided the antibodies against CD248. And that's it. Thank you.
Questions for Dr. Conway? Dr. Lopez.
You said that CD248 increases the activity of tissue factor, and you also mentioned in your summary that in whole blood. So do you find it in blood?
Yeah, there are soluble fragments of CD248 in blood, in whole blood. How it gets there, I don't know.
So one thing that I was thinking was that it's possible that the CD248 can change the membrane microdomain localization of tissue factor, and in that way change its activity possibly, and also perhaps increase the likelihood that it will be blebbed off in a viral-like particle, a microparticle, and go into the blood.
Yeah, I mean, it's a reasonable thought. We haven't looked at that at all, and it's an interesting idea. Yeah, I don't know that. Yeah.
It's the story about cholesterol and fats and so on is all up in the air. Should I be getting my CD48 measured?
Well, CD248 measured, and right now, we don't know, we're only measuring mRNA levels, transcripts in the subcutaneous fat, because we don't have an ELISA to see if the levels in blood have any relationship to the expression in the fat tissue. So I don't know the answer, but that would be a very interesting little, tool to have in terms of insulin, identifying particularly risk of insulin resistance. Yes. We have them lined up. Go.
Ed, so what if you were to block CD248 with an antibody or inject it into fat? Do you see any conversion in
Okay, so the question is about blocking it. All right, so we did some studies, the obesity studies, because we had access to some antibodies, but we have no idea how those antibodies work. We know where they're targeted, but we have no idea what they do, and so we saw no effect. So in just an obesity study, a fat diet. So that doesn't tell us anything. It would be great if it worked, but it didn't. It didn't do anything. And we're doing studies now in terms of functional assays to test whether the antibodies have any effect on the functional activity of tissue factor. Yeah.
Ed, there was a guy, I don't remember his name, who was claiming that the fat metabolism and was due to an adenovirus. Is there any reality to that?
Ooh, I have absolutely no idea.
Yeah, see, there were several papers on that. Some kind of adenovirus.
Yeah, I don't know. Anybody know? Don't know.
I don't know.
Kathy? No?
No, he had several publications. I think he even has a company to study that.
Okay, I'll look that one up.
And he said that obesity was due to a viral attack. So
That's a good excuse. Great. I like that.
I think so too.
Any other questions?
We're good? Oh, Jose.
Have you looked, or are you planning to look at the impact of changes in diet on the levels of the CD248?
You're talking about in people now.
Or animals? I mean, you could do it, you could look kind of in an epidemiology kind of way in people, but you could actually at least do the experiment in animals.
Yeah, great idea. It wasn't our first thing because we were really working hard to try to figure out the mechanism by which the coagulation part is working. But certainly it would be great to see dietary changes. I'm sure now we will, particularly with the Karolinska group, because they collect a lot of these patients at varying levels of obesity.
That will also answer the adenovirus question.
Exactly. Okay. All right, good.
Thank you very much.
Thank you.
Get off the stage. That was a great first half of the afternoon. We've got a break. Please be back here at 4:14. We'll start back at 4:15.